network approach and using pathways and signatures. This time, we also, over the course of that 10 years, got more samples, more clinically annotated samples. So we had 100 samples to train our classifier on, and then a completely separate set of 78 samples on which to test it. So remember, I talked about unsupervised as well as supervised learning. So here's the unsupervised learning. So we took our 96 samples and we clustered them according to their expression. And what we found were 6 subtypes within the full class of things that by a doctor were called meduloblastomas. And this is just the biological annotation or characterization of those different subtypes. And if this were a biology audience, I would probably go into this a little bit more in detail. But basically what you can see, even looking here, is that different genes are amplified or there are more copies of different genes in some of the subtypes, that some pathways are activated in some of the subtypes, and so on. So they're different at the molecular or the biological level. And that's not something you would see under the microscope. And if you look at, this is what's called a Kaplan-Meier curve. So what happens here is over time, from 0 to 180 months, you're seeing the number of patients that have died over that time period, the number of children that have died. So what you can see, for example, is that this green subtype, or C4, has the best prognosis. And notice it also has the most, it has a lot of male, well, it's about evenly divided male and female. The worst prognosis case is this C1 case. That actually has 4 times as many males. We don't really understand why that's the case, but little boys are overrepresented in that case. But you can see that they're very different survival rates for these different subtypes. So given that, it's clear that we want different predictors, right, for these different subtypes, because they're different. They're molecularly different. So that's what we do. And yeah, this is just saying C1 drives C4 outcome. If you look at it as a single disease, the outcome varies by subtype, and yeah, and this is association, not prediction. Okay, so we're going to use a knowledge-based Bayesian network model, and what do I mean by that? To do the prediction, we're going to estimate the probability of relapse from clinical and genomic features. We'll use a predefined network structure. The features are associated with clinical outcome come from published studies, analysis of our training data from the 96 samples that we had to train on, and we're going to use pathway and gene set and signature features rather than individual genes to increase the signal to noise. And what you'll see is that I will show you a collection of nested submodels where at each stage we increase the number of features that we use, or the number of variables that we use, okay? And we increase the types of features. And then we're going to emphasize this subtype. Okay. So I want to go from individual gene space to signature space. So I have to have a way of projecting from this space of 20,000 genes, and I'm going to project actually into a space of about 2,600 gene sets. Curated pathways that are related to cancer, and oncogenic, that means causing cancer, gene sets. So how do I do that projection? Well, I do that projection in a similar way, a sort of Kolmogorov-Smirnov-like way, that I did the gene set enrichment score. But in this case, I'm doing it to a single sample, so I don't do any expression differential expression. I don't sort the genes according, because I only have one sample, so I can't, sort genes according to their correlation with outcome, because I only have a single sample and I don't know what it is. So in this case, I just sort the genes according to their absolute expression. So the highest expressed genes at the top, the lowest expressed genes at the bottom. And then the formula here is very similar. It's a slightly smaller coefficient that we use. In fact, in this case, the coefficient is about a quarter. And in this case, instead of using the maximum deviation from 0, we use the integral under this enrichment score or random walk plot. Okay. But it's very similar, and the main difference is it works on one sample at a time. So these are the kinds of features that I'm going to use. Again, I'll use clinical features, pathway features, whether the pathway has a high or low activation score, as determined by this projection. What is this not doing what I want it to do? Okay. Sorry, I'm in a little technical trouble. Okay, so this is how I get those activation scores, right? And then the subtype, they're going to be subtype dependent features. Remember I said we're going to do different things for each subtype, and then we're going to use markers of the subtype to distinguish. So when I get a sample, I have to first figure out which of the 6 subtypes it's in. Then I'm going to use in the model to make the prediction some features that are independent of its subtype and some features that are dependent on its subtype. And I'm going to use clinical features, expression features, copy number features, and so on. And I'm going to discretize that activation signature. And to do that, I'm just going to use a greedy split based on the contingency tables. And I split here so that the errors on both sides are equal. That's a little detail which is not all that. Okay, so this is what the outcome model looks like. All right. So the first model, remember I said they were nested models that added types of features and numbers of features. So the first one just depends on the clinical attributes of histology, which is what does the sample look like under the microscope, and metastasis, whether it's spread from the brain. Model B adds a subtype independent signature, which is CMAC activation. So CMAC is an oncogene, that is, it's a cancer-causing gene, and if it's activated, that's a signature that corresponds to poor outcome. Right? But this is a feature that was not derived from the data, so it wasn't trained on the data, but rather just extracted from the literature. It's been known for a long time that CMAC activation is related to poor outcome. Here's where the meat is. So over here in model C, we add the subtype. So first we determine the subtype based on expression signatures, 6 for each disease subtype, in a similar way to the way that we did it with individual genes, except remember, we're projecting into that space of signatures, the 2,600 signatures, and doing it there. Then we add disease subtype dependent expression signatures. So these are the poor and good outcome signatures that are related to each of the subtypes. So one for subtype C1, one for C2, one for C3, one for C4, one for C5, one for C6. And then finally, in model D, we take all of these things and we add something about copy number gains and losses, right? So that's the copy number, remember, is the number of copies of a particular gene in the cancer cells. And here again, you see that CMYC coming up and NMYC, and then a deletion of chromosome 6, the Q arm of chromosome 6, and here's some other ones. So these three, the ones with the red dot, come from the literature. These four were trained on the data. So it's a combination of the two things. So how do we do? This is the model. The model itself just is a simple Bayesian log odds model, right? Nothing fancy here. And the features, as we said, and the auxiliary model to predict subclass, which is also log odds evidence. So here are the results. And this is actually quite nice. Because notice that this was just clinical, right? This was the subtype independent model. And you see that here's train and test. Remember, these were not trained on the data. So they don't do all that well. But once you get the subtype involved in the model, then you get a significant increase in the accuracy of the model. See that big jump. And then in Model D, you lose a little bit of accuracy. Why do you lose it? We think you lose it a little bit because we didn't have enough data actually to train on in that case. We didn't have copy number for all the data. But the other thing I want you to notice here is that the difference between train and test is very small, which means we're not overtraining the model. So that's a good thing. And here are just the numbers that correspond. Here's the training set accuracy, and here's the test set accuracy. And you can see how close they really are. But here's the other thing, is that our model rescues 6 out of 15 patients. What do I mean by rescues? So if you just use the clinical model, which is what's used by doctors today, then 15 patients were clinically classified as standard risk. But of those 15 patients, they were classified as standard risk, but they went on to relapse. So the clinical classification of these 15 patients was you're a standard risk patient, but within 3 years, these children relapsed. We rescued 6 of them. Namely, we said 6 of these patients are actually high-risk patients. So 40% of the patients, remember, these are little kids, that for whom the prognosis was incorrect, we predicted a correct prognosis. And that's an important thing because that means they're candidates for a little bit more intense therapy. This is just another way of looking at where you can see that as you add the subtype, you do much better in prediction. But this is really what I wanted to show you, which is this is a picture of the amount of evidence we have for the samples in the training set. And this tells you whether there are samples that relapsed, but that's the black, and that didn't relapse, that's the white, that's the clinical relapse data. But what I want you to look at is over here, you see this is the copy number data, and you see how much copy number data is missing. So we have a lot of expression data, but not much copy number data. And remember I showed you that we lost a little bit of accuracy when we needed to. So how do you get this kind of information into the clinic? How do you I can't go into a physician's office and start explaining Bayesian models to them. So how do we move this kind of approach into the clinic and into the practice of medicine? Well, the idea is to use what's called a nomogram. So here's an example of a nomogram. So suppose you were on the Titanic before it sank. What were the odds of your surviving? on the Titanic. All right. Well, let's look. If you're prior, so that means before we take into account anything about you, is that you're not going to survive. Right? That's what this red, this is not surviving. But if you look here, what you can see is that you have more of a chance of surviving if you're a child than if you were an adult. If you were a female, you had more of a chance of surviving than if you were a male. And lo and behold, if you were in first class, right, you had a much better chance of surviving. If you were in second class, well, maybe you could do okay. Here's the sort of neutral point. If you were in third class, no chance, right? So, if you were a little girl in first class, you did very well and you survived. So, but this is an easy way for someone to look at the different pieces of evidence that go into determining or predicting your survival from this accident. So can we do that with this model? And the answer is yes. So here's an example of a, I think this is a high risk, a standard risk patient with respect to the clinical data. Let me see if I can find the clinical data. Yeah. So down here is the clinical data, and this is a standard risk patient here. But if you look at the features that we're using in the model, and then you look at the log odds, you see that we predict this to be, right, a relapse patient, so a high-risk patient. But this is something that a physician could sort of look at and see what the different genomic features are that are adding to that risk thing, rather than showing them equations. And our plan, or our hope, is actually to deliver it on an iPad. You know, the patient, when the resection is done of the tumor is sent out to the lab, the tests are made, the expression and the copy of the tests are made, and the physician gets back a little report that was. So that's the hope. And this is just some of the work we've been doing on ovarian cancer using the same approaches, liver cancer, myelodysplasia, follicular lymphoma, and so on. We're trying to get, instead of using signatures from the literature and so on, we're actually in the lab now trying to extract better signatures of pathway activation and dysregulation by actually doing them in different cell types in the lab and then taking those measurements. We're also adding DNA information, so looking at mutations in the tumor's DNA and adding that as another set of features to improve the predictive capability, and then the idea of using continuous features and multivariate features. We hope to improve the accuracy. So again, how do we make our sophisticated methods available and accessible to biologists? Biologists traditionally have not been very analytical, not been very computational. That makes them good biologists, but sometimes makes communication back and forth a little difficult. So can we get these techniques into their hands in an easy way? And so a part of my lab spends its time taking the methods and approaches that the other part of my lab develops and turning them into software that's user-friendly and accessible that any biologist can use. And also to provide tools for visualizing all this different kind of genomic data to help to generate hypotheses for further study in the lab. So one of the big projects on the software side of the lab is this infrastructure for computational genomics. We call it, it's called Gene Pattern. It's freely available on the web. And the idea here is that a biologist can sit at a computer and in a very friendly, menu-driven, point-and-click way, access lots of sophisticated methods and data sets and integrate them into workflows, analytic workflows that they can then share with other people. And it keeps an automatic history so that it can be reproduced by anyone. And so when you get to publish, when you get to the point of publication, you have the whole computational workflow there that can be repeated by anyone. The other thing, this is probably just a little bit more about the software. This is what the interface of the software looks like. These are all the methods that we include in it. And as you can see, it's used by a lot of people all over the world. We also run a server, which is not mentioned here, which runs, actually this number is out of date. I think there's more like 30,000 users now. But many of them take advantage of a public server that we use that runs 2,000 to 3,000 jobs a week. The gene set enrichment analysis that I spoke about in that molecular signatures database is also freely available. It has even more users, and we're constantly adding signatures. And when the experimental work is done that I spoke to you about, that will also go into this database. This is a visualization tool which has been extraordinarily popular in the community. It allows you, I'm sorry that you really can't see this. It allows you to look at multiple types of data in the same view. And is really helpful at targeting genomic, what we call genomic aberrations in data. And again, to help generate hypotheses. And again, I apologize for the lack of color here. And then finally, a much more recent project is to take a number of tools, 6 popular tools in the community, 2 of which come from my lab, but the other 4 come from other labs, and this is a project that runs from California to Israel. Many institutions are involved, and we have some funding from the NIH, but also Amazon Web Services because it lives in the Amazon cloud. And the idea here is that you can very easily send data back and forth between these tools, and they make it easy for the biologists because they don't have to do any transformations of the data themselves. It's all done automatically. And we drive it by these 3 different biological projects, one of which is the medulloblastoma patient stratification project that I just spoke to you about. So that's sort of the software side of what we do. And none of this work, of course, can be done without lots and lots of collaborators. The Broad Institute is a very collaborative and open institute where biologists, mathematicians, computer scientists, engineers, physicists all work together trying to address these difficult problems. And so I end on this note, which is that really, I think, to solve the problems facing us in biomedical research today, it's going to not just take biology and the wet lab, but it's also going to take mathematicians, statisticians, computer scientists, all working together to really tackle these problems and to bring the promise of genomics into the clinic. And for myself, I was trained as a mathematician. I worked in nonlinear analysis and for many years, and I made this change into biology about 20 years ago. And I think there's nothing more satisfying than to think that the work that I do is really going to make a difference for a little 2 or 3-year-old who comes into the clinic to be treated for an invasive brain tumor. That's not, proving theorems was great. I did it for many years, but for me, the satisfaction of using the knowledge and the techniques that we have, which are extraordinarily powerful on this kind of data, and we now have the ability to acquire large amounts of data and analyze them and pull out all kinds of signatures. And so this work in cancer that I do, also in malaria and HIV, these are going to have impacts on real people and their lives. And so I encourage all of you to think about the possibilities here and to maybe get involved. in some way or another. Thank you very much.
Thank you, Jane, for your wonderful talk. Any question? Comment?
What is the biggest challenge when you want to teach machine? And in here, you need a new program, new algorithm for the machine, or is something else?
You know, the biggest challenge in this kind of work is the data itself. The data is extraordinarily noisy. And the fact that we're moving towards acquiring the data from sequencing helps a little bit because it's a little bit more, it's more discrete. But even then, there are so many problems with the actual technique, experimental techniques of sequencing, and the noise in the data makes it very, very difficult. The other difficulty, if you're doing this kind of clinical work, is that physicians are not very precise in their descriptions of patients and their conditions. So, without telling tales out of school, one of my clinical collaborators in this medulloblastoma project would come back week after week, having modified the clinical annotation of the samples, until, we finally said, okay, go home, lock yourself in a room for 2 weeks, and do not come out until you've finished correctly annotating these samples, because, mistakes in the annotation lead to mistakes in the models. So I think it's really the extraction of signal to noise in the noisy data and in the annotation itself. But these, in terms of data mining and machine learning, the annotation of the data issue is something that people deal with all the time. American Express, for example. That's why sometimes your credit card gets canceled even though you were using it legally. Yeah, it's happened to me also. What do you mean you're in Vietnam?
So, I don't understand what you gave example of cancer on time. Is that different for other type or for other type or
Every type of cancer needs a slightly different model, yes. Because the mechanisms which give rise to different cancers can be different. There are some similarities across cancers. So there are certain oncogenes or tumor suppressors that will occur. So their dysregulation problems with them will be seen in multiple kinds of cancer types. But there are other things which are unique to specific cancers. So in this medulloblastoma, for example, the sonic hedgehog gene is very particular to that type of cancer. Whereas the tumor suppressor, it's called P.2, its dysregulation occurs in many different cancers.
So for other disease?
Yes.
Not cancer, but like food effect. More.
For other diseases?
Yeah, like food effect or pressure.
Yeah, so a lot of the work, so I look at molecular data, functional data, I call it functional data, molecular data, this is gene expression and copy data. For cardiovascular disease, most of the work that has been done has been on what we call germline DNA. So the genetic predisposition for disease. And what makes things like cardiovascular disease there are genetic influences on disease, but there are also environmental influences on disease. So if you're looking at causality, then it's important for something like heart attacks to look at your germline DNA, your actual DNA, your genome, to see whether you have predisposition. But then there are lifestyle issues, do you eat too much fat? Do you smoke? Do you not do enough exercise? You know, all those things. So that's a little different.